you should think about process development and research development a little bit differently compared to how you do that in biologics or small molecules. I think that aspects of process development should be included and incorporated much earlier in the drug development process for an ATMP, just to make it more successful, to reduce the rate of failure, and also to ensure that you don't have to repeat a lot of stuff later on. The second thing is just a reminder, and that is ATMPs are much, much more complex, but also much more costly to develop. So people are often surprised by how expensive just the development aspect is. That is something that you should be aware of when you do that. And the last thing is probably the early decision making.
Welcome back to the Smart Biotech Scientist podcast. I'm David Brillman, your host, and we are continuing our deep dive into cell and gene therapy development with Oliver Kraemer, a technical development leader at Flagship Pioneering. In part 1, we explored the fundamental differences between biologics and ATMPs. Now, we are tackling the nitty-gritty of cell and gene therapy development. Let's jump right back into the fascinating conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brillman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
I would like to touch upon yet another difference between especially cell therapies and biologics. For ATMPs, actually it's the process and the biology set the boundaries. What does this mean?
So if you talk to classical biologics development people, what they usually say, what the mantra is, the process is the product. And that is true for biologics. So however you produce your product, the quality of the biologic that you generate will be impacted. I think this boundary aspect that I use in there is more important for ATMPs, specifically for cell therapies, because the process really, really does define what you will see for a cell therapy. There is a concept of biological fitness that is, I think, something that is probably underrepresented. And it talks about the way how cells behave in culture, very roughly speaking. So even if you start with a monoclonal culture, there is a certain variability in your culture. And every cell looks maybe just a tiny bit different compared to the other cell. It could be a different regulation. Some pathways are more active than others. Not much, but just a little. And from classical population dynamics, we know that when we change the environment that these cells are in, that some of these cells will have a growth advantage or a different selective advantage than others. So if you do your development in a certain setting, the cells experience a certain setting, and then you get a certain defined output of your quality, of your efficacy, of your yield, whatever you're measuring. But when you then change that environment, that could be, for example, switching from a 2D to a 3D system, or changing the production media because your research media is not available in GMP, then you will likely get a slightly different cell type that you end up with. And in an ideal world, there is not much change that you will see. But in the more common cases that we have seen, you do see differences. You see differences in yield, you see differences in quality, and that impacts your product significantly, which also talks then a little bit about the offset between development data and research data that you have to think about. And for some of these cells, I have evaluated quite a few of them, research lines, commercial lines, all of those. And we see that certain things we want to do with them don't necessarily work well with all of them. So, for example, some grow beautifully in a 2D system, differentiate well if you talk about iPSCs, and behave very textbook-like. But then when you try to transition them to a 3D system, for example, they fail. They become genetically unstable, or they just don't grow that much. That's something that happens, and that's this boundary aspect that I'm talking about. Not everything is easily achievable with every cell line. And therefore, you have to think about, when do I test these things, and how do I actually want to bring something forward to the market or to the clinic?
So there is a tremendous complexity when you're developing cell therapies. You have a lot of moving parts to take care of. I mean, there's an interdependence of the cell line, the media, the process. Yeah, you said whether it's a 2D system, a 3D system. So there's a lot of things to manage. So this leads me to this question. So how do you ensure alignment between research results, the clinical trials, and then ultimately the final product? Because, I mean, giving all this complexity and the duration of the projects, you have a whole lot or many things to manage.
Yes, that's true. And the answer to that is similar to the problem, very complex. We have chosen an approach that I understand might not be doable for every company in every situation. But for example, for the team I'm working with, or the company I'm working with, we do not really have a differentiation between research team and development team. So we have all kinds of different expertises mixed into one team. So we don't really differentiate between the research aspect of the project and the development project, as it goes all hand in hand. If you do this in a more classical setting, I think it is important that all relevant parties discuss this ahead of time. What is relevant? What do we have to show? How do we have to show it? And how do we ensure that this is then translatable? Similar to the cell line question I spoke earlier, there has to be a discussion. Do we want to maybe do an evaluation of the cell line pretty early on? Or do we delay this? Or how do we want to proceed with that? How do we want to generate the amount of cells that we would like to test in our animal models or in our in vitro tests? That all needs to be discussed. Even though it seems simple, that is just not the usual practice necessarily, because in biologics, this is less important. Not saying that it's unimportant, but it is less important because you have predefined packages that you can deliver. And that is just not the case for ATMPs. Now, if you talk about the interconnectedness between media, cofactors, cell line, process, that is a very big challenge. And there is unfortunately no easy answer to that. In a best case scenario, you would have unlimited resources and just put everything into one giant DOE and just test everything that you think impacts that. But we all know that is very unlikely to happen. So you have to break that down in smaller chunks and see that you evaluate certain aspects first and then validate them with the subsequent aspects of your process and your product.
So to make this now specific, let's talk about, for instance, the dependence between the cell line and the media. When would you do that, and how would you do that to actually to find something that works and you will not need to change later down the way?
So the way we are doing this and have done this is we actually, when we evaluate a cell line, look at different media that have a path forward to GMP or to commercial very early on in the project. and we actually test the cell line in different medias and just see how it reacts and how it grows, how stable it is, if we see genetic aberrations, if we see changes in pluripotency, for example, if it is an iPSC line. And honestly, we just test that. That can be done in relatively small scale. So it is not like you have to have a full-blown large-scale process development setup for that. That is something that you can test in a smaller scale. But it is important to think about it and keep in mind that you have to test this. So we only test things that have a path forward that we could end up with using in a GMP or clinical setting. And that has so far turned out quite good for us.
Cell therapies and also gene therapies, they have a huge potential and they enable us to get access to life-saving therapies. Just a recent example with the sickle cell disease. I mean, a game changer, and there's a lot of other things happening on the cell therapy side. What needs to happen today so that more people get access to these therapies? Because the costs are still very high. It's still very difficult to develop these therapies. What would you like the industry to change so that we can serve a much wider range of patients?
Good question. One of the things that I think is important for this is the question of autologous versus allogeneic. I am, of course, aware that there are certain challenges with allogeneic product. You can have immunogenicity. Sometimes you have less efficacy of the product. Those are things, however, that you can work on and can develop. If you look at the cost distribution, and a lot of that is the COGM part, so cost of goods for manufacturing, you will see that as soon as you go away from the one patient, one dose production scheme, you have much more favorable conditions also for the patients. So that translates directly to patient access because your therapy can be manufactured per dose cheaper. I think that is something that is very important. So pushing forward towards more allogeneic cell therapies would enable that greatly. Now, if you think a lot about gene editing, even for cell therapies or as a therapy, that is, of course, also still very expensive. So every investment into making your production process more efficient will help to drive down these costs. So those are things that the industry is actively working on, but it is a challenge right now. But I think that these are two things that can help patient access and can reduce the cost.
And would you see some kind of revolutionary approach or a different approach that would enable us to develop much faster and way better and reduce the risk of failure?
I think the reducing the risk of failure is something that will likely come with experience. So I don't think that there is a magical thing where you just have an epiphany and know suddenly, oh, if I do this, the outcome will be great. I think it will be similar to what we have experienced in biologics. It takes a while until we have learned what we can do, what we maybe should avoid, and how we can make things more efficient and better. Now, what I do think is beneficial is if you take these learnings from the biologics world and apply them to your ATMP world. There are a lot of things that are transferable and that you can leverage there. And I think we should do that.
Yeah, I totally agree. We have learned so much in the biologic fields over the last 4 years. I think there's a lot of things we can leverage. Obviously, yes, there are things that are different and we have to not just leverage it, but perhaps transform it or transpose it. But yes, there's a lot of experience we can't leverage.
Fully agree.
And I think also we should find ways to connect people who are working in the biologics field to people who are working in the cell and gene therapy space. I feel there is still two different silos or it's different worlds, but I think we should much more work together and leverage the expertise we have and the technologies we have at our disposal.
Yes, I completely agree. And that's something that I have seen as well. If you look at I'm not sure if I can say classical ATMP development because again, it's not that old, but a lot of experience in ATMPs, cell therapy, like iPSC therapy, but also gene editing comes right now directly from the academic field. And a lot of the ways that these processes are developed are academic, and they differ significantly from what you do in industry. And I do think that when we connect people from the biologics field much closer to this ATMP knowledge, that there is just a different angle of how to look at a problem and how to think about it that will be beneficial for that development. And I think it is starting right now. There are more biologics aspects that slowly get into the world of AT development and ATMP manufacturing. But I do think it is something that we should continue to push for.
Yeah, absolutely. We should definitely facilitate this process. Now, as we are wrapping up, Oliver, what is the most important takeaway you want our listeners to walk away from our conversation with?
Yeah, I think I have 3 basic key takeaways that are dear to me, and I think that would help people that think about developing an ATMP product. One is that you should think about process development and research development a little bit differently compared to how you do that in biologics or small molecules. I think that aspects of process development should be included and incorporated much earlier in the drug development process for an ATMP, just to make it more successful, to reduce the rate of failure, and also to ensure that you don't have to repeat a lot of stuff later on where time is critical. The second thing is just a reminder, and that is ATMPs are much, much more complex, but also much more costly to develop. So people are often surprised by how expensive just the development aspect is. That is something that you should be aware of when you do that. And the last thing is probably the early decision making. I do think that some decisions in ATMP development have to be taken a lot earlier compared to biologics. We talked about cell line selection, maybe process design, maybe your scale question, what are you actually aiming for to supply later on? And that changes a little bit how you develop this product. And those 3 things, I think, are the main takeaways for me.
Wow, thank you very much, Oliver, for these amazing takeaways. Thank you for sharing the insights. And smart biotech scientists, why don't you just hit the pause button and rewind and listen to these 3 takeaways again? They will help you develop. Thank you once again, Oliver, for sharing all your expertise, and thank you for being on the show today. And where can people connect with you?
Yeah, absolutely. It was a pleasure. I think most easy is actually LinkedIn. I am available on LinkedIn, so just reach out and connect with me.
Fantastic. Yes, please reach out to Oliver. I will leave the link down in the show notes. And thank you once again, Oliver, for being on the podcast today.
Thank you very much. It was a pleasure.
What a fantastic journey through the complexities of cell and gene therapy development. A huge thank you to Oliver Kraemer for sharing his wealth of knowledge and practical insights. If you are looking to strengthen your CMC development strategy, I'm offering you a focused strategy workshop to evaluate and refine your approach with precision. Ready to take your development process to the next level? Book a free call with me. You will find the link in the show notes. Until next time, this is David Brulman signing off.
All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, Visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.